Somewhere in the future, a child will be born who has never felt Earth's gravity. Not on a launch pad, not in low orbit, inside an artificial habitat, drifting through interstellar space, surrounded by metal walls and recycled air, and a silence no human infant has ever known. That child will grow up breathing manufactured atmosphere, walking on floors curved by rotation, building bones and muscles and balance under conditions no human body was ever designed for. And here is what should unsettle you. If that child ever traveled to Earth, the planet that created our entire species, it might crush them. Their heart might fail, their bones might fracture, their lungs might struggle against an atmosphere that feels impossibly heavy. The first true children of deep space may never survive the world that made us. If you find these kinds of deep explorations interesting, take a second to hit like and subscribe. It means more than you know. Now, get yourself comfortable. Let's begin. Every civilization tells a story about its first child. The first baby born on a new continent. The first infant delivered in a colony, across an ocean. These births carry symbolic weight because they represent permanence. A people did not merely visit a place. They stayed long enough to bring new life into it. That act transforms exploration into settlement. One day, if humanity survives long enough and reaches far enough, there will be a first child born not on any continent, not on any planet, not on any moon, but inside a machine. A vessel built by human hands, drifting through the emptiness between stars, carrying the species forward into a darkness no generation before has ever called home. That moment will be celebrated. It will be historic. It will be broadcast back to Earth on a signal delayed by years or decades, and people will cry and cheer and call it the beginning of something extraordinary. But almost nobody is talking about what that birth actually means for the body of that child. Because here is a fact that should keep you awake. Despite more than 6 decades of continuous human spaceflight, despite sending hundreds of astronauts to orbit for months at a time, despite building and operating the International Space Station for over 25 years, humanity has never once witnessed a human pregnancy in space. No conception, no embryonic development, no fetal growth, no birth. Not a single data point exists for how a human being forms from a fertilized cell into a breathing infant anywhere other than on the surface of this planet, shielded by this atmosphere, held together by this gravity. We have sent humans into space. We have never grown one there. That distinction matters more than almost anyone realizes, and it separates everything we think we know about human survival in space from the question that will define whether our species can truly leave Earth behind. When most people imagine humans living in deep space, they picture astronauts floating aboard a spacecraft, fit, trained professionals conducting experiments and gazing at the stars. Maybe they picture a rotating station with artificial gravity, something from a science fiction film with families walking along curved corridors and children playing in parks that wrap upward along the walls. These images are comforting. They are also dangerously incomplete, because they skip over a question so fundamental that it threatens every assumption about human expansion into the cosmos. Can a human body develop normally if it has never experienced Earth, not visit Earth, not return to Earth after a mission, but never, from the very first moment of cellular division, exist under the gravitational, radiological, atmospheric, and biological conditions of this planet? Every human who has ever lived was formed inside a body standing on Earth, shaped by a gravity field pulling at 9.8 meters per second squared, shielded by a magnetosphere blocking the worst of cosmic radiation, surrounded by an atmosphere rich in oxygen and teeming with microbial life. Every bone in your skeleton formed under that pull. Every chamber of your heart learned to pump blood against that resistance. Every structure in your inner ear that tells your brain which way is up calibrated itself to that specific force. You are a product of Earth in every cell, every tissue, every system. You were built by this planet whether you think about it or not. Now imagine removing all of that, not reducing it, removing it, or replacing it with something else entirely. An artificial spin producing a fraction of Earth's pull, a sealed habitat with recycled air, a radiation environment that no magnetic field fully blocks, a microbial ecosystem containing only what humans deliberately brought aboard. That is what born in deep space actually means. Not born on Mars, where at least a partial gravity field of roughly 0.38 g exists. Not born on the Moon, where 1/6 g still provides some gravitational stimulus. Born inside an artificial structure, far from any natural gravitational body, where the only forces acting on a developing human are the ones engineers designed into the vessel. And here is what transforms this from a theoretical exercise into a genuine scientific crisis. We have almost no data on what that would do to a developing human body. Almost none. The research that does exist is startlingly thin. Most of what we know about human biology in space comes from studying adult astronauts on missions lasting 6 months to a year aboard the International Space Station. These are fully developed adults, people whose skeletons finished growing decades earlier, whose cardiovascular systems calibrated to Earth gravity in childhood, whose immune systems trained on a lifetime of terrestrial microbial exposure. When these adults enter microgravity, their bodies begin to change. The changes are measurable, significant, and in some cases alarming, but they are changes to finished systems, modifications to structures that already formed correctly. That is a fundamentally different situation from a body that never formed under Earth conditions in the first place. Let me show you how dramatic those adult changes are, because they set the stage for understanding why developmental changes could be far worse. When an astronaut arrives at the International Space Station and begins living in microgravity, their skeleton starts dissolving. That is not hyperbole. Bone remodeling is a constant process in the human body. Specialized cells called osteoblasts build new bone tissue, while other cells called osteoclasts break it down. On Earth, mechanical loading from gravity and movement keeps this cycle in balance. The skeleton senses the forces placed upon it and maintains enough density to bear those loads. Remove the loading, and the balance collapses. Osteoclast activity accelerates. Osteoblast activity slows or stalls. The result is bone loss at a rate of approximately 1 to 2% per month in weight-bearing bones like the pelvis, spine, and legs. The European Space Agency has documented cases where astronauts lost up to 20% of bone mineral density in weight-bearing bones after a 6-month mission. That rate of loss is comparable to the bone deterioration seen in elderly patients with advanced osteoporosis, except it happens in otherwise healthy individuals within months rather than decades. A study published in 2022 tracked 17 astronauts using high-resolution imaging of their tibius and found that 12 months after returning to Earth, 9 of those 17 still had not fully recovered their bone density. The researchers described the loss as equivalent to a decade or more of age-related bone deterioration compressed into a single spaceflight. Another study, published in late 2024, confirmed that the window for bone recovery after spaceflight is limited. Most targeted bone reformation occurred in the first 6 months after landing. After that, the recovery process largely stalled. The structural connections within the spongy interior of the bone, once broken, did not magically reconnect. New bone formed on the remaining scaffolding, but the architecture itself was permanently altered. This is what happens to adult bones that already finished developing. Fully mineralized, structurally complete, weight-tested skeletons that spent decades building themselves under Earth gravity. Even these robust structures cannot fully recover from 6 months without gravitational loading. Hold that thought. Muscles follow a similar trajectory. Without the constant resistance of gravity, skeletal muscles begin to atrophy almost immediately. The legs and lower back are hit hardest, because these are the muscles that work the most against gravitational pull on Earth. Astronauts aboard the ISS exercise for approximately 2 hours every day using specialized resistance equipment, and they still lose significant muscle mass. Without exercise countermeasures, the loss would be catastrophic. Studies have shown decreases in muscle volume, changes in muscle fiber composition, and reductions in peak force output, all within weeks of arriving in microgravity. The cardiovascular system undergoes its own dramatic restructuring. On Earth, your heart works constantly to push blood upward against gravity toward your brain. The muscles in your legs squeeze veins to help return blood from your lower extremities back to the heart. In microgravity, this entire system becomes unnecessary. Blood no longer pools in the legs. Instead, body fluids shift upward toward the head and chest. Astronauts develop the characteristic puffy face and skinny legs appearance within hours of reaching orbit. The heart, no longer needing to pump as hard against gravity, begins to weaken and shrink. Studies have found that the left ventricle of the heart can become more spherical after extended time in microgravity, reducing its pumping efficiency. Orthostatic intolerance, the inability to maintain stable blood pressure when standing upright, is one of the most common problems astronauts experience when they return to Earth. Some cannot stand for more than a few minutes without feeling faint. Then there is the vision problem. NASA identified a condition now called Spaceflight-Associated Neuroocular Syndrome, or SANS, after discovering that a significant percentage of astronauts returning from long-duration missions had measurable changes to their eyes. Globe flattening, optic disc swelling, shifts in visual acuity. The leading hypothesis is that the headward fluid shift in microgravity increases intracranial pressure, pressing on the optic nerve and altering the shape of the eyeball. Research published in 2025 confirmed that SANS features develop in 38 to 51% of astronauts on long-duration flights. Some of these changes persist after landing. For certain astronauts, vision never fully returns to pre-flight levels. The immune system degrades in space as well. Research from multiple agencies has documented broad dysfunction across both innate and adaptive immunity during spaceflight. White blood cell populations shift. The body's ability to detect and fight infection weakens. The gut microbiome, the vast community of beneficial bacteria that plays a critical role in immune regulation and overall health, changes in composition and loses diversity. A study from the University of California, San Francisco, found that microgravity abnormally activates T regulator cells, suppressing immune responses that the body would normally mount against infection. The overall pattern is one of chronic, low-level immune dysfunction that leaves astronauts more vulnerable to illness precisely when they re-enter an environment teeming with microbes. Every one of these effects has been confirmed through decades of observation, measurement, and peer-reviewed study. Bone loss, muscle atrophy, cardiovascular deconditioning, vision changes, immune suppression. These are not theoretical risks. They are documented realities of human spaceflight, and every single one of them describes what happens to an adult body, a body that already developed fully, a body that already built its bones under gravitational loading, a body whose heart already learned to pump against 1G, a body whose vestibular system already calibrated to Earth's pull, a body whose immune system already trained on decades of microbial exposure. These systems were finished before the astronaut ever launched. They degrade in space, yes, but they degrade from a completed state. The architecture was already there. The foundation was already laid. Now consider what happens when there is no foundation. We have almost no human data for this scenario because no human has ever developed in space. But we have a small and deeply unsettling collection of animal studies that hint at what might happen. In 2023, a team of Japanese researchers published a landmark study in a peer-reviewed journal describing the first successful culture of mammalian embryos aboard the International Space Station. Frozen 2-cell mouse embryos were launched to the station aboard a SpaceX Falcon 9 rocket, thawed in orbit, and grown for 4 days under both microgravity and artificial 1G conditions produced by an onboard centrifuge. The embryos developed to the blastocyst stage, the critical early cluster of cells that would normally implant in a uterine wall and begin forming a fetus. The researchers found that gravity did not appear to significantly block blastocyst formation. This was widely reported as encouraging news for the possibility of mammalian reproduction in space, but the details were less reassuring than the headlines suggested. The number of viable blastocysts recovered from the microgravity group was small. Up to 25% of the microgravity blastocysts showed a structural abnormality where the inner cell mass, the cluster of cells that would eventually become the actual fetus, was split into 2 separate locations rather than gathered into one coherent mass. In the ground control group, this splitting occurred in only 6 to 7% of embryos. The researchers themselves cautioned that the sample size was too small to draw firm conclusions, but noted that gravity may be important for the inner cell mass cells to properly organize. This is one experiment. It covered only the first 4 days of development, roughly 1/4 of a mouse's 20-day gestation period. The embryos were not transferred into living mice. No fetus was grown, no birth occurred. No pup was raised to adulthood. The study demonstrated that early cell division can proceed in microgravity. It did not demonstrate that a mammal can develop normally in space. Earlier research filled in additional pieces of this puzzle, none of them comforting. A separate study found that when mouse embryos were fertilized and cultured under simulated microgravity on Earth using a three-dimensional clinostat, the birth rate of offspring transferred to recipient mothers was less than half that of the 1G control group. The embryos that did develop showed slower growth and fewer trophectoderm cells, the outer layer that forms the placenta. Another study exposed pregnant mice to spaceflight during mid to late gestation and found modest but measurable effects on birth rate, litter size, birth weights, and neonatal mortality. A different study on a shuttle-era mission reported developmental issues in the brains of mouse pups born after their mothers experienced microgravity during pregnancy. In one of the earliest attempts to study mammalian reproduction in space, male and female rats were flown in orbit with the goal of observing mating behavior in microgravity. The rats did not mate. The animals appeared confused and disoriented, unable to coordinate the physical mechanics of reproduction in an environment without consistent gravitational orientation. The experiment was considered a failure from a reproductive standpoint. No conception occurred. Meanwhile, studies of luteal cells, the ovarian cells responsible for producing progesterone to maintain early pregnancy, showed increased DNA damage, decreased progesterone secretion, and increased cell death when cultured under microgravity conditions. Research on oocyte maturation found that the rate of egg cell development dropped from 73% under normal gravity to under 9% under simulated microgravity. The organization of the meiotic spindle, the cellular structure essential for proper chromosome separation during egg cell division, appears to be gravity-dependent. Taken together, these studies paint a portrait of reproduction that is far more fragile than most people imagine. Fertilization might occur in microgravity, early cell division might proceed, but every subsequent step, implantation, placentation, organogenesis, fetal growth, birth, neonatal survival, remains either untested or troubled by the limited data that exists. No mammal has ever completed its entire reproductive cycle in space. No mammalian offspring has been conceived, gestated, born, and raised to reproductive adulthood in microgravity. The full experiment has never been run, and here is what makes this gap so dangerous. Every plan for long-duration human habitation in deep space, every generation ship concept, every interstellar colony proposal, assumes that this experiment will work. It assumes that human beings can reproduce successfully away from Earth. It assumes that children can be conceived, carried to term, delivered safely, and raised to healthy adulthood under conditions radically different from those in which our species evolved. These assumptions underpin every serious proposal for making humanity a multi-planetary or interstellar civilization, and they rest on almost no evidence. Human pregnancy is currently contraindicated during spaceflight. That is not a guideline. It is a safety prohibition enforced by every major space agency. NASA, the European Space Agency, and other agencies explicitly prevent pregnant astronauts from flying. The reason is simple. The risks to a developing fetus are unknown and potentially catastrophic. No agency is willing to accept liability for an outcome they cannot predict or control. This prohibition is itself a confession. It acknowledges that we do not know what space does to developing humans, and that our ignorance is so profound that the safest course is to prevent the situation entirely. It is a policy born from the absence of data, not the presence of reassurance. But policies cannot hold forever. As missions grow longer and move farther from Earth, the question of reproduction will become unavoidable. A 3-year round trip to Mars creates a window long enough for pregnancy to occur, planned or otherwise. A generation ship traveling for decades or centuries to another star system requires reproduction by definition. You cannot sustain a population without it. At some point, humanity will have to confront this gap in knowledge directly. And when it does, the question will not simply be whether a healthy baby can be born in space. That is the first question, the minimum threshold, the baseline for everything else. The deeper question, the one this entire story is about, is what kind of body that baby will have. Whether a human being whose skeleton, muscles, heart, brain, inner ear, eyes, lungs, and immune system all developed under fundamentally different conditions will be the same kind of human that Earth produces, or whether they will be something new, not a different species, not a mutant, not an alien, but a human being whose body was built by a different set of physical rules, shaped by a different gravitational environment, tuned to a different world. Because there is a critical difference between adapting to space and developing in space. Every astronaut health effect we have documented, every bone density scan, every muscle biopsy, every cardiovascular assessment, every immune profile describes what happens when a fully formed Earth body adapts to a new environment. The body was already built. The blueprint was already executed. The house was already standing when the storm hit. Some walls cracked, some foundations shifted, but the structure existed before the stress arrived. Developing in space means building the house during the storm. It means laying the foundation without knowing what forces it will need to bear. It means constructing a skeleton without the gravitational signal that tells bone cells how thick to make the walls. It means growing a heart without the resistance that teaches cardiac muscle how hard to pump. It means wiring a vestibular system without the consistent gravitational input that defines which direction is down. It means assembling an immune system inside a sealed container with a microbial ecosystem containing only a tiny fraction of the organisms the human body evolved to encounter. This is not adaptation. This is formation, and formation is a one-way process. The developmental windows during which bones mineralize, muscles establish their fiber composition, neural pathways wire themselves to sensory inputs, and organs calibrate their function to environmental conditions are finite. Many of them close permanently in childhood. A vestibular system that never calibrated to 1G during fetal development and early childhood may never be able to function under 1G at all. A skeleton that never experienced full gravitational loading during the growth years may never achieve the density needed to support an adult body on Earth. A heart that never built the muscular strength to pump blood against a full gravity field may never develop the capacity to do so. These are not speculative fears. They are reasonable inferences drawn from confirmed developmental biology. We know that mechanical loading drives bone formation in growing children. We know that gravitational input shapes vestibular calibration from the fetal period through adolescence. We know that cardiovascular conditioning during development establishes the baseline function that the heart will maintain for life. We know these things from decades of pediatric medicine and developmental research conducted entirely on Earth. What we do not know is what happens when these developmental signals are absent, reduced, or replaced by something artificial. And that uncertainty represents the single largest unaddressed risk in humanity's entire plan to become a spacefaring species. We have spent billions of dollars developing rockets, habitats, life support systems, and propulsion technologies. We have mapped potential destinations. We have designed radiation shielding and food production systems and water recycling plants. But we have invested almost nothing in answering the most fundamental question of all. Can the human body develop normally away from Earth? The answer might be yes. It might be that artificial gravity, radiation shielding, carefully managed nutrition, and advanced medical care can replicate the essential conditions of Earth closely enough that a child born in deep space would develop indistinguishably from a child born in Kansas or Kyoto or Nairobi. That is the optimistic scenario, and it is entirely possible. But it is also entirely unproven. And if the answer is something other than yes, if development in deep space produces a human body that is subtly or profoundly different from an Earth-developed body, then the implications reach far beyond medicine. They reach into the future of the species itself. Because a human being whose body formed under different conditions might thrive in the habitat that shaped them. They might live a full, healthy, extraordinary life aboard a rotating station or inside a generation ship. But they might find that the planet their species came from, the world their ancestors called home, the only natural environment in the known universe that produces human beings, has become a place their body cannot survive. If gravity shapes adult bodies this dramatically in just 6 months of spaceflight, imagine what it does, or fails to do, to a body that has never known gravity at all. That question leads somewhere that should make every future planner, every rocket engineer, and every dreamer of interstellar civilization deeply uncomfortable. Because the answer does not depend on how clever our technology becomes. It depends on something far older and far less forgiving. It depends on the biology of how a human being is built. And that building process, as you are about to discover, is far more dependent on gravity than anyone expected. You do not think about gravity. You have never had to. It has been there since the moment your body began assembling itself from a single fertilized cell, pressing on every tissue, pulling on every fluid, loading every structure with a force so constant and so universal that your biology treats it the same way your lungs treat oxygen, not as something extra, as something essential, a condition so fundamental to how you were built that removing it does not simply make life harder. It changes what kind of body gets built in the first place. This is where the science becomes genuinely unsettling, because gravity is not just a force that holds you to the floor, it is a developmental instruction, a signal that tells your bones how dense to become, a cue that tells your heart how strong to grow, a constant input that calibrates one of the oldest and most critical sensory systems in the human body, one that most people have never even heard of. And every single one of these processes depends on gravity being present during specific windows of development that once closed, never reopen. Start with bone, not adult bone, not the finished skeleton of an astronaut losing density in orbit. Start with the skeleton of a developing human being, because the process of building bone is fundamentally different from the process of maintaining it. When a fetus develops inside the womb, its skeleton begins as cartilage, soft, flexible tissue that provides a rough scaffolding for the body's shape. Over the course of gestation and childhood, this cartilage is gradually replaced by true bone through a process called ossification. Specialized cells called osteoblasts deposit minerals, primarily calcium and phosphate, onto the cartilage framework, transforming it into hard, dense bone tissue. This process does not happen randomly. It follows a principle that orthopedic medicine has understood for more than a century, sometimes called Wolff's Law. Bone adapts to the loads placed upon it. Apply mechanical stress to a bone, and the bone remodels itself to become stronger in the direction of that stress. Remove the stress, and the bone weakens. It sheds density it no longer needs. On Earth, the most consistent and pervasive mechanical load acting on the developing skeleton is gravity. From the moment an infant begins to support its own head, every interaction with the gravitational field sends signals through the skeletal system. Crawling loads the arms and wrists. Standing loads the spine, pelvis, and legs. Walking, running, jumping, falling, each of these childhood activities transmits forces through bone tissue that trigger osteoblasts to strengthen the skeleton in precisely the areas that need it most. The process continues throughout childhood and adolescence, peaking in early adulthood when bone mineral density reaches its lifetime maximum. After that, the body shifts to maintenance mode. The architecture is set. The density is established. The skeleton is built. This is critical. The density and structural quality of your adult skeleton were determined largely by the mechanical loading your bones experience during growth. Peak bone mass, the maximum amount of bone your body ever builds, is influenced by genetics, nutrition, hormonal factors, and physical activity during childhood and adolescence. But physical activity in this context means activity against gravity, running on a playground, jumping off a swing, walking to school. Every step a child takes on Earth is a gravitational loading event that tells the skeleton how strong it needs to become. Now remove gravity from that equation, not reduce it, not replace it with some fraction of Earth's pull. Remove it entirely, as would be the case aboard a vessel in deep space without artificial gravity, or replace it with a partial simulation produced by rotation. What happens to a skeleton that never receives the full gravitational loading signal during its developmental years? We do not know with certainty because the experiment has never been conducted on humans, but we can reason from what we do know. We know that adult astronauts lose 1 to 2% of bone density per month in microgravity, even with aggressive exercise countermeasures. We know that much of this loss is never fully recovered. We know that the structural connections within trabecular bone, the spongy interior scaffolding, once broken during unloading, do not reconnect when gravity returns. And we know that adults are losing bone they already built. Their skeletons had decades of gravitational loading before spaceflight began, They had a full reservoir of density to draw from. A child developing in microgravity or low gravity would have no such reservoir. Their bones would never receive the full loading signal. The osteoblasts would never get the instruction to build to Earth standard density. The skeleton would form, yes. Cartilage would ossify. Bones would grow in length and shape. The genetic blueprint for a human skeleton would still execute. But the density, the internal architecture, the load-bearing capacity of that skeleton would be calibrated to whatever gravitational environment the child actually experienced. If that environment provided only a fraction of Earth's gravitational pull, the skeleton would build itself to handle a fraction of Earth's gravitational pull. Think of a tree that grows inside a greenhouse where no wind ever blows. The tree will grow. It will have a trunk and branches and leaves. But the trunk will be thin and flexible because wind is the mechanical signal that tells a tree to thicken its trunk and strengthen its wood. Transplant that greenhouse tree into an open field with real wind, and it will snap. Not because it is a defective tree, because it was built for a world without wind. The architecture that a windswept tree develops, the dense wood, the thick bark, the deep root system, was never triggered in the greenhouse. The developmental window passed. The tree became what its environment told it to become. A human skeleton developing in low gravity faces the same fundamental problem. The bones will form, but they will form for the world they actually experience. And if that world provides a quarter of Earth's gravity, or a tenth, or none at all, the skeleton that results may be entirely functional in that environment and entirely inadequate for Earth. Not weak in the sense of being diseased, weak in the sense of being correctly built for a different set of physical rules. Now, move from bone to muscle. The logic is similar, but the timeline is even more compressed. Skeletal muscles develop their fiber composition, their neural recruitment patterns, and their baseline strength during childhood through constant interaction with gravitational resistance. Every time a child lifts their head, pushes themselves off the floor, climbs a step, or carries an object, they are training their muscular system against gravity. The specific types of muscle fibers that develop, the ratio of slow-twitch endurance fibers to fast-twitch power fibers, and the neural pathways that coordinate muscular contraction are all shaped by the mechanical demands placed on the body during growth. In microgravity, those demands vanish. An infant born in 0G would never need to lift its own head. It would never crawl against resistance. It would never pull itself upright against a downward force. The postural muscles of the back and legs, the muscles that on Earth are among the most constantly active in the body, would have no reason to develop the strength or endurance they would need to function under 1G. The body would still build muscles. The genetic program would still produce fibers and attach them to tendons and bones. But the calibration of those muscles, their strength, their composition, their neurological integration, would reflect an environment where gravitational resistance does not exist. Adult astronauts who return from 6-month missions struggle to walk for days or weeks. Their legs feel like rubber. Their sense of balance is disrupted. Their muscles must relearn how to support their body weight. And these are individuals whose muscles developed fully on Earth. They spent decades building the neuromuscular architecture for upright locomotion under 1 GeG. They are readapting to a system they already knew. A person who never built that architecture in the first place would not be readapting. They would be attempting to operate a system their body never constructed. The difference between relearning to walk and learning to walk in a body that was never designed for walking under 1G is not a difference of degree. It is a difference of kind. But the most profound developmental dependency on gravity may not be bones or muscles at all. It may be the system most people have never consciously thought about, the vestibular system. Deep inside each of your inner ears lies a set of structures so small they could fit on your fingertip, yet so essential that without them you could not stand, walk, focus your eyes during head movement, or even understand which direction you are falling. The vestibular system consists of 3 semicircular canals and 2 otolith organs, the utricle and the saccule. The semicircular canals detect rotational movement of the head. The otolith organs detect linear acceleration and the direction of gravitational pull. Together, they provide the brain with continuous, real-time information about the body's position and motion relative to gravity. This is not optional information. It is the foundation upon which nearly every other motor function is built. balance, posture, gaze, stabilization, spatial orientation, coordination, the ability to navigate through a three-dimensional world without falling over. All of these depend on vestibular input. When the vestibular system fails or degrades, the consequences are devastating. Chronic vertigo, inability to maintain balance, nausea, disorientation, inability to read or focus the eyes during any head movement, loss of spatial awareness severe enough to make walking dangerous. And here is the fact that should alarm anyone thinking about human development in deep space. The vestibular system is one of the very first sensory systems to develop in the human embryo. The structures of the inner ear begin forming as early as the 3rd week of embryonic development, when thickened patches of surface tissue on either side of the embryonic hindbrain start to fold inward, creating the structures called otic placodes. By the 5th week, the semicircular canals are taking shape. Hair cells, the sensory receptors that detect motion and gravity, begin differentiating between the 11th and 13th weeks. The vestibular nerve begins myelinating, developing its insulating sheath that allows rapid signal transmission around the 20th fetal week. It is the first cranial nerve to complete myelination. By the 24th week of gestation, when the inner ear development is essentially complete, vestibular ganglion cells have reached their mature form. By the 32nd week, the vestibular system is functional enough to produce a fully developed Moro reflex, the startle response that causes a newborn to fling out its arms when it senses a sudden change in support. That reflex is a direct sign of vestibular brainstem integration. The vestibular system is not merely early to develop. It is early to develop because it needs to be. It provides the foundational sensory input around which much of the brain's motor programming organizes itself. From birth onward, every movement a child makes is interpreted through vestibular information. Tummy time, rolling over, sitting up, crawling, standing, walking, running. All of these developmental milestones involve the vestibular system, telling the brain how the body is oriented relative to gravity, how fast it is accelerating, and what adjustments the muscles need to make to maintain balance. This calibration is not instant. It extends through childhood and into adolescence. Research on pediatric vestibular development indicates that proprioceptive integration, the ability to combine vestibular input with body position sense, matures around ages 3 to 4. Visual weighting and balance, the ability to properly integrate what you see with what your vestibular system reports, reaches adult levels around age 12. Full vestibular maturity, the complete integration of vestibular, visual, and proprioceptive systems into a seamless balance network, may not occur until ages 15 to 17. This means the vestibular system spends roughly the first 17 years of a human life calibrating itself to the gravitational environment. 17 years of constant input from Earth's 1G field, shaping neural pathways, tuning sensitivity thresholds, training the brain to interpret gravitational acceleration as the baseline against which all other motion is measured. Now, imagine a child whose vestibular system develops not under 1G but under a fraction of it, or under the simulated gravity produced by a rotating habitat, which introduces a Coriolis effect that creates different sensations depending on the direction and speed of head movement, or under no consistent gravitational pull at all. That child's vestibular system would still develop. The genetic program would still build semicircular canals and otolith organs and vestibular neurons. But the calibration of that system, the sensitivity settings, the neural pathways, the reflexive motor responses, would be tuned to whatever gravitational environment the child actually experienced. A musician who has never heard the instrument they are meant to play can still learn the finger positions, read the sheet music, and understand the theory. But when they finally hear the instrument for the first time, nothing they practiced in silence prepares them for the reality of sound. The vestibular system of a child raised in fractional gravity might understand balance in a way that functions perfectly inside a rotating habitat. But Earth's full gravitational pull would be a sensory input their system was never calibrated to process. The result would not be a simple adjustment period. It could be permanent disorientation, chronic vertigo, an inability to maintain upright posture under a force the brain has no framework to interpret. This is not speculation built on nothing. This is inference built on confirmed developmental biology. We know that vestibular calibration depends on gravitational input during critical developmental periods. We know that those periods have finite windows that close in childhood and adolescence. We know that adults whose vestibular systems are damaged or disrupted experience severe, often permanent impairment. A child whose vestibular system calibrated to the wrong gravitational signal would not have a damaged system. They would have a system that works exactly as designed, for a world that is not Earth. The cardiovascular implications follow a similar developmental logic, though the specifics are different. On Earth, the human heart develops inside a fetus that is itself inside a mother standing, sitting, and moving under 1G. From the moment the fetal heart begins beating, at roughly the 4th week of gestation, it is pumping blood through a body that exists within a gravitational field. After birth, the heart must immediately begin working against gravity. Blood must be pushed upward to the brain. Venous return must be managed against gravitational pooling in the lower extremities. The baroreceptors, the pressure sensors in the blood vessels that regulate heart rate and vessel constriction, calibrate themselves to the hydrostatic pressure gradients created by 1G. Throughout childhood, the cardiovascular system continues to develop and strengthen. The heart grows larger and more muscular. Blood vessels develop the tone and elasticity needed to manage pressure distribution under gravitational loading. The autonomic nervous system refines its ability to make rapid adjustments, increasing heart rate when you stand up, constricting vessels in the legs to prevent blood pooling, dilating vessels in the brain to ensure adequate perfusion. Every one of these adaptations is a response to living under gravity. A heart that develops in microgravity or low gravity would never receive these signals. It would never need to pump blood upward against a strong gravitational pull. It would never develop the muscular wall thickness of a heart that spent its formative years working against 1G. The baroreceptors would calibrate to a pressure landscape defined by an environment without significant hydrostatic gradients. The blood vessels would develop tone appropriate for a system where gravitational pooling does not occur. What would happen if that person then travelled to Earth? Based on what we know about adult cardiovascular deconditioning in space, the answer is concerning. Adult astronauts who spent months in microgravity struggle with orthostatic intolerance so severe that some cannot stand without assistance for days after landing. Their hearts have weakened, their blood vessels have lost tone, their baroreceptors are miscalibrated. And these adults spent decades building cardiovascular fitness under 1G before their missions began. They have a baseline to return to. A person whose cardiovascular system developed entirely in low gravity would have no baseline. Their heart would not be deconditioned. It would be correctly conditioned for a world with less gravitational pull. Bringing that heart to Earth would be like asking a sedan engine to do the work of a truck engine. Not because the sedan engine is broken, but because it was built for a different job. Research into SANS, the neuroocular syndrome observed in astronauts, adds another layer. The ocular and cranial changes caused by SANS persist in some astronauts even after returning to Earth, evidence that the fluid redistribution in microgravity leaves a lasting structural imprint. If those changes occur in adults whose cranial anatomy already formed under 1G, imagine the developmental consequences for a fetus whose brain, eyes, and cranial vasculature form in an environment where fluid distribution follows entirely different patterns. The skull itself might develop a different shape. The optic nerves might form under different pressure conditions. The venous drainage pathways from the brain might calibrate to a pressure environment that does not exist on Earth. These are not trivial cosmetic differences. They are structural variations in the organ that defines human consciousness. But the brain is not merely a passive recipient of fluid dynamics. It is itself a structure that builds its functional architecture in response to gravitational experience. And this may be the most consequential developmental dependency of all, because while bones can be braced and muscles can be supported and hearts can be assisted with medication, the brain's internal wiring determines how a person moves, navigates, balances, and inhabits physical space. If that wiring develops without the gravitational input it expects, the consequences reach into every physical action the person will ever attempt. The human brain does not arrive at birth with a finished map of the body. It builds that map through experience. The motor cortex, the strip of brain tissue responsible for planning and executing voluntary movement, organizes itself during infancy and childhood through a process driven by sensory feedback. When a baby reaches for an object, the motor cortex sends a signal to the arm muscles. The arm moves. Sensory receptors in the muscles, joints, and skin send information back to the brain about what happened. Did the hand reach the object? Did the arm overshoot? How much force was required? The motor cortex uses this feedback to refine its commands, gradually building a detailed internal model of the body's capabilities and limitations. On Earth, every one of these feedback loops operates against gravity. Reaching upward requires more force than reaching sideways. Lifting an object requires the brain to calculate how much muscular effort is needed to overcome both the object's weight and the gravitational pull on the arm itself. Standing up from a seated position requires coordinated activation of dozens of muscle groups, all calibrated to the specific resistance that 1G provides. The motor cortex builds its internal model of the body by interacting with gravity thousands of times per day, every day, from birth through adolescence. By adulthood, this model is so refined that you can reach for a glass of water without consciously calculating the trajectory. The computation happens automatically, built into neural circuits that gravity helped create. In microgravity, or fractional gravity, those feedback loops would carry fundamentally different information. Reaching upward would require no more force than reaching sideways. Lifting an object would require overcoming only inertia, not weight. Standing would not require the same muscular coordination because the gravitational load driving the need for that coordination would be absent or greatly reduced. The motor cortex would still build an internal model of the body, but that model would reflect a world where movement costs less energy, where objects have inertia but negligible weight, where the body does not need to constantly fight a downward pull. The neural circuits that emerge from this process would be optimized for that environment and potentially bewildered by the demands of 1G. The cerebellum adds another layer to this problem. This dense structure at the base of the brain is responsible for motor coordination, timing, and the fine-tuning of movement. It is the part of the brain that allows you to walk smoothly rather than lurching from step to step, to catch a ball rather than swatting at the air where the ball was a moment ago, to maintain your balance on uneven ground without conscious effort. The cerebellum develops its extraordinary precision through years of practice, integrating input from the vestibular system, the eyes, and the proprioceptive receptors throughout the body into a seamless coordination network. Critically, the cerebellum develops much of this precision during childhood. The period of most rapid cerebellar growth and synaptic refinement overlaps with the period when children are learning to crawl, walk, run, climb, and manipulate objects. Every one of these activities provides the cerebellum with calibration data, and every one of them on Earth occurs against the resistance and within the framework of gravitational force. A child learning to walk is not merely learning the sequence of leg movements. They are learning how to manage a controlled fall in one specific gravitational field, how to shift their center of mass in response to one specific downward pull, how to time their muscle contractions to counteract one specific acceleration. The cerebellum records all of this and uses it to build the coordination programs that will govern movement for the rest of that person's life. A child whose cerebellum calibrated to a different gravitational environment would have coordination programs built for that environment. Their movements might be fluid, precise, and beautifully coordinated inside their habitat, but those same programs would produce errors under 1G. Reaching for an object would use too little force. Walking would involve timing miscalculations because the speed at which the body falls forward during each step depends on gravitational acceleration. Catching a thrown object would require recalculating trajectories that the cerebellum learned under different physics. The body would not feel clumsy in the way that a tired person feels clumsy. It would feel calibrated to the wrong world. Like a cartographer who has never seen the territory they are mapping, the brain would have built exquisitely detailed movement programs for a place it has never been. Spatial cognition, the brain's ability to understand and navigate three-dimensional space, compounds the problem further. Humans develop spatial awareness through interaction with an environment where gravity provides a permanent, reliable reference direction. Up is always up, down is always down. The brain uses this gravitational vertical as the anchor for its entire spatial framework. Rooms are oriented relative to the floor, which is always the direction gravity pulls. Objects are understood as being above, below, beside, or behind based on a gravitational coordinate system so deeply embedded in cognition that most people never realize it exists. In a rotating habitat, the gravitational reference is not truly vertical, it is centripetal, directed outward from the axis of rotation, and it is not uniform. Moving through the habitat changes the strength and direction of the apparent gravitational pull, depending on proximity to the axis. A child growing up in this environment would develop spatial cognition anchored to a reference frame that does not exist on Earth. Their brain's model of space itself, the intuitive understanding of which direction things fall, how trajectories curve, and how the body relates to its surroundings, would reflect the physics of a rotating cylinder rather than the physics of a planet. On Earth, that spatial model would produce constant, low-level confusion. Not the dramatic vertigo caused by vestibular mismatch, but a subtler and perhaps more persistent sense that the world does not behave the way the brain expects it to. Objects would fall differently. Rooms would feel oriented incorrectly. The spatial grammar the brain uses to interpret its environment would be speaking one language while Earth speaks another. The cumulative neurological picture is this. A space-developed brain would contain a motor cortex mapped to the wrong gravitational resistance, a cerebellum coordinated to the wrong physics, a spatial cognition system anchored to the wrong reference frame, and a vestibular integration network calibrated to the wrong input signal. None of these systems would be damaged. Every one of them would be performing precisely the task it was trained to perform. But the training happened in the wrong classroom, under the wrong physics, against the wrong forces. And unlike a broken bone that can be reset or a weakened muscle that can be retrained, a neural architecture, wired to the wrong gravitational physics, cannot simply be overwritten by exposure to the correct one. Each of these systems, bone, muscle, vestibular, cardiovascular, develops under the assumption that gravity is a constant, not a variable, not an option. A constant. The genetic code that builds a human body does not contain instructions for life in 0G or fractional g. It contains instructions that interact with environmental inputs to produce a body suited to the conditions it actually encounters. On Earth, those conditions include a gravitational field of 9.8 meters per second squared, acting uniformly in one direction at all times. Remove that input, and the genetic program still runs. A human being still forms, but the body that results is a human being shaped by a different world. This is the crucial distinction that separates the challenge of deep space birth from every other challenge in human spaceflight. Rocket engineering is hard but solvable. Life support systems are complex but designable. Radiation shielding is expensive but improvable. But the developmental dependency of the human body on gravity is not an engineering problem. It is a biological reality etched into the deepest layers of how our species builds itself. You cannot patch it with better technology. You can only try to replicate the gravitational signal closely enough that the body develops as if it were on Earth. And that is where artificial gravity enters the conversation. Not as a solution, but as an attempt at one. The most commonly proposed mechanism for artificial gravity in space is rotation. Spin a habitat fast enough, and the centrifugal effect pushes everything inside toward the outer wall, creating a force that mimics gravitational pull. The concept is well understood in physics. Gerard O'Neill described rotating cylindrical habitats in the 1970s. NASA studied them seriously. The mathematics work. A cylinder roughly 8 kilometers in diameter, rotating at about 28 revolutions per hour, would produce a force equivalent to 1g on its inner surface. But the engineering reality is daunting. No rotating habitat of any meaningful size has ever been built in space. The structural materials required to construct a cylinder kilometers in diameter that can withstand continuous rotational stress do not yet exist at scale. The energy required to spin up and maintain rotation is enormous. The logistical challenge of building such a structure in deep space, far from any manufacturing base, exceeds anything humanity has ever attempted. And even if such a habitat could be built, it would not perfectly replicate Earth's gravity. Rotation introduces the Coriolis effect, a force that acts perpendicular to the direction of motion within the rotating frame. Walk in the direction of rotation inside a spinning habitat and you feel heavier. Walk against it and you feel lighter. Move your head and the fluid in your semicircular canals behaves differently than it would under true gravity. Throw a ball and it curves in ways that defy terrestrial intuition. The smaller the radius of rotation, the stronger these effects become. A habitat with a radius of only a few dozen meters would produce Coriolis forces strong enough to cause nausea, disorientation, and difficulty with motor coordination. Even at larger radii, the effect never fully disappears. For an adult who grew up on Earth, the Coriolis effect in a rotating habitat would be disorienting but manageable. The brain would adapt because it already has a calibrated vestibular baseline to work from. But for a developing child, the Coriolis effect would be part of the gravitational signal itself. The vestibular system would calibrate to a world where down is not truly down, where moving your head produces sensations that have no equivalent on Earth, where the apparent force of gravity changes depending on which direction you walk. That calibration might produce a vestibular system that functions beautifully inside the rotating habitat and fails catastrophically under the uniform, non-rotating gravitational pull of an actual planet. There is also the gravity gradient to consider. In a rotating habitat, the effective gravitational force decreases as you move closer to the axis of rotation. On the outer floor of the habitat, you experience full simulated gravity. Move halfway to the center and you experience half. At the central axis, you experience weightlessness. A child growing up in such a habitat would exist in a world where gravity changes with altitude. Their body might develop different bone densities and different limbs depending on how much time they spent at different distances from the outer wall. Their cardiovascular system might learn to handle a range of gravitational loads rather than one consistent pull. This adaptive flexibility might serve them well inside the habitat. It might also leave them uniquely unprepared for a planet where gravity is uniform, inescapable, and much stronger than anything they experienced at the center of their rotating home. The honest scientific assessment, based on everything we currently know about developmental biology and spaceflight physiology, is that we do not know the minimum gravitational signal required for normal human development. We do not know whether partial gravity, say the 0.38 grams of Mars, is sufficient to produce a skeleton, cardiovascular system, and vestibular apparatus capable of tolerating 1 GeD. We do not know whether artificial gravity produced by rotation introduces developmental distortions that would impair function under true gravity. We do not know whether any technological solution can fully replace the developmental role of a natural gravitational field. What we do know is that every system in the human body that depends on gravitational input for its development represents a potential point of divergence between an Earth-born human and a space-born human. And the list of such systems is long: bones, muscles, heart, blood vessels, inner ear, eyes, brain fluid dynamics, postural reflexes, motor coordination, even the proprioceptive system, the body's sense of its own position and movement, develops through interaction with gravitational resistance. Each of these systems develops during finite windows, miss the window, and the calibration is set. The tree grows for the greenhouse it lives in. The body builds itself for the world it knows. And gravity is only one variable in the equation. The habitat that shapes a deep space child's body also exposes that child to threats that Earth's natural systems have shielded every previous human from. Threats that compound the developmental risks already created by altered gravity and introduce entirely new categories of danger. Because beyond the gravitational question lies another set of forces acting on every cell in a developing body, invisible, penetrating, and in deep space, relentless. On Earth, you are shielded by an invisible fortress you have never seen and will never feel. It extends tens of thousands of kilometers into space, wrapping the planet in a magnetic cocoon that deflects the worst radiation the cosmos produces. Behind that magnetic barrier sits a second layer, an atmosphere dense enough to absorb most of what makes it through. Together, these two systems have protected every organism on this planet for billions of years. Every cell that has ever divided, every embryo that has ever formed, every child that has ever grown, did so under the shelter of a radiation environment so mild that your body does not even register it as a threat. In deep space, that fortress does not exist. And for a developing human, the consequences of its absence are not theoretical. They are cellular, molecular, and potentially catastrophic. The radiation environment beyond Earth's magnetosphere is dominated by two primary sources. The first is galactic cosmic rays, a constant background flux of extremely high-energy particles that originate outside our solar system. These particles, primarily protons but including heavier ions like helium nuclei and fragments of iron, carbon, and oxygen, have been accelerated to nearly the speed of light by supernovae, neutron star collisions, and other violent astrophysical events. They stream through interstellar space in every direction, and they are extraordinarily difficult to stop. Unlike the relatively gentle radiation environment inside Earth's magnetic field, galactic cosmic rays carry enough energy to penetrate several meters of solid material. They pass through spacecraft hulls. They pass through human tissue. When they strike an atom inside a cell, they can strip electrons, shatter nuclei, and leave behind a trail of ionization damage that stretches across the interior of the cell like a microscopic bullet wound. The second source is solar particle events. These are bursts of charged particles ejected by the sun during solar flares and coronal mass ejections. They are less energetic per particle than galactic cosmic rays but can arrive in massive quantities over short periods, delivering dangerous doses of radiation within hours. On Earth, the magnetosphere deflects most of these particles, funneling them toward the poles where they produce the aurora. In deep space, there is no such deflection. A solar particle event striking an unshielded or lightly shielded spacecraft would expose everyone aboard to a concentrated pulse of ionizing radiation. The International Space Station operates within Earth's magnetosphere in low Earth orbit, where the magnetic field still provides substantial protection. Astronauts aboard the ISS receive elevated radiation doses compared to people on the ground, but those doses are moderated by the geomagnetic shielding. A deep space habitat operating between Earth and Mars, or beyond the solar system entirely, would receive the full, unfiltered flux of galactic cosmic rays plus whatever solar particle events the Sun produces. Measurements taken by instruments aboard NASA's Mars Science Laboratory during its transit to Mars recorded radiation doses of approximately 300 to 350 millisieverts during a 180-day journey. For context, the general public on Earth receives roughly 2 to 3 millisieverts per year from natural background radiation. A 6-month transit to Mars exposes an astronaut to more than 100 times the annual terrestrial dose. Now consider what this means for a developing embryo. Ionizing radiation damages biological tissue primarily by breaking chemical bonds. When a high-energy particle strikes a molecule of DNA, it can sever the double helix, mutate base pairs, or cause chromosomal rearrangements. Adult cells have sophisticated repair mechanisms that can fix many types of DNA damage, though imperfectly. Some damage gets misrepaired, introducing mutations that may lead to cancer years or decades later. But adult tissue is mostly stable. Cells divide relatively slowly. The genome in each cell has been replicated and error-checked many times. The stakes of any single DNA break, while real, are distributed across billions of cells that are not actively dividing. Embryonic and fetal tissue is the opposite. It is defined by rapid cell division. From the moment of fertilization, cells are dividing at extraordinary rates, doubling and redoubling to build every organ, every tissue, every structure in the body. During organogenesis, the period when the basic body plan is laid down, a single cell error can cascade into structural abnormalities affecting entire organ systems. The first month after fertilization is the most vulnerable period. Cells are dividing so rapidly and differentiating into so many specialized types that damage to even a small number of cells can have outsized consequences. A high-energy cosmic ray striking an embryo during this period could destroy cells, critical to the formation of a limb, an organ, or a neural structure. Because the embryo is tiny, often smaller than a grain of rice during the earliest stages, a single particle track could traverse a significant fraction of the entire developing body. Research has established that radiation exposure exceeding 100 millisieverts during pregnancy is associated with elevated rates of congenital abnormalities, growth impairment, premature delivery, cognitive disability, and miscarriage. This threshold was established from studies of populations exposed to radiation on Earth, including survivors of atomic bombings and communities near nuclear accidents. The radiation involved in those cases was primarily gamma rays and neutrons, forms of radiation that differ in some respects from the galactic cosmic rays encountered in deep space. Galactic cosmic ray particles, particularly the heavy ions, produce far more concentrated damage along their tracks than gamma radiation does. A single ion nucleus traveling at nearly the speed of light leaves a trail of ionization so dense that the cells along that track are often destroyed outright, while the surrounding tissue absorbs secondary radiation from the particle fragments and displaced electrons. This concentrated damage pattern means that the biological effectiveness of galactic cosmic rays, the amount of biological harm caused per unit of energy deposited, is substantially higher than conventional radiation. The exact factor depends on the particle type and energy, but estimates suggest that heavy ions can be 5 to 20 times more damaging per unit dose than gamma rays. This is a critical distinction because it means that the radiation doses measured during deep space transit, already high by terrestrial standards, understate the biological impact on developing tissue. As pregnancy progresses, the risks shift but do not diminish. The growing fetus presents an increasingly large target. The probability that a cosmic ray particle will strike the uterine wall, the placenta, or the fetus itself increases simply because there is more tissue to hit. Research has identified a specific concern with the uterine muscle. If a cosmic ray strikes myometrial tissue, the smooth muscle of the uterus, it could trigger localized contractions. A sufficient number of such interactions could initiate premature labor. On Earth, premature delivery is survivable with intensive neonatal care. In deep space, hundreds of millions of kilometers from the nearest hospital, premature birth could be fatal. Shielding against galactic cosmic rays is extraordinarily difficult. Unlike solar particle events, which consist primarily of lower energy protons that can be stopped by relatively modest material barriers, galactic cosmic rays include particles so energetic that they penetrate meters of solid material. Worse, when these particles strike the atoms in shielding material, they can produce secondary radiation. The original particle shatters the nucleus of a shielding atom, producing a shower of neutrons, protons, and other fragments that scatter into the habitat interior. In some cases, the secondary radiation produced by shielding is more biologically harmful than the original particle. This means that simply piling more mass around a habitat does not necessarily reduce the radiation dose to occupants. There is an optimal shielding thickness beyond which additional material actually increases the dose. Hydrogen-rich materials like polyethylene and water are more effective than metals because hydrogen nuclei are close in mass to the incoming protons and absorb their energy more efficiently, producing fewer dangerous secondary fragments. But even the best shielding materials cannot fully block galactic cosmic rays. They can reduce doses, sometimes substantially, but they cannot eliminate them. Some proposals involve magnetic shielding, using powerful superconducting magnets to generate an artificial magnetosphere around the habitat. Theoretical models suggest this approach could significantly reduce radiation exposure, but no such system has been tested at scale in space. The engineering challenges of generating and maintaining magnetic fields powerful enough to deflect cosmic rays, estimated at 5 to 20 Tesla, around a habitat are formidable. The honest assessment is that any human living in deep space will receive elevated radiation exposure compared to living on Earth. Shielding can reduce the dose. It cannot eliminate it. For adults, this elevated exposure translates to increased long-term cancer risk, a serious concern but one that manifests over decades. For a developing embryo, fetus, or young child, the same exposure occurs during the most radiation-sensitive period of human life. The cells are dividing faster. The repair mechanisms are less mature. The consequences of unrepaired DNA damage are more severe, because each damaged cell gives rise to thousands or millions of daughter cells that carry the same mutations forward. This creates a grim calculus. Every day a developing child spends in deep space is a day of elevated radiation exposure during the window, when their biology is most vulnerable to it. The cumulative dose over a full pregnancy and childhood could be substantial even with aggressive shielding, and the biological effectiveness of the deep space radiation environment means that each unit of dose carries more risk than the equivalent dose on Earth. But radiation is only one dimension of the biological isolation that defines life in deep space. There is another system in the human body that depends entirely on environmental exposure during development, one that receives almost no attention in discussions of space colonization, but may prove equally consequential. The immune system. Your immune system was not born knowing how to protect you. It was trained. From the moment you passed through the birth canal, your body was colonized by microorganisms. Bacteria from your mother's body seeded your gut, your skin, your respiratory tract. Over the following months and years, you encountered thousands of microbial species in your food, your water, the soil you played in, the air you breathed, the animals you touched, and the other humans you lived among. Each encounter was a lesson. Your immune system learned to recognize pathogens and tolerate beneficial organisms. It built a library of immune memories, antibodies, and cellular responses calibrated to the specific microbial world you inhabited. By adulthood, your body hosted trillions of microorganisms, collectively called the microbiome, that play essential roles in digestion, vitamin synthesis, immune regulation, and even neurological function. This training process is not optional. Children who grow up in excessively sterile environments show higher rates of allergies, autoimmune disorders, and hypersensitivity reactions. The hygiene hypothesis, now supported by extensive research, demonstrates that reduced microbial exposure during childhood impairs the development of immune tolerance, the ability to distinguish genuine threats from harmless substances. The immune system needs exposure to a diverse microbial world during development. Without that exposure, it becomes either overreactive, attacking the body's own tissues or harmless environmental substances, or underreactive, failing to mount adequate responses to genuine infections. Now consider the microbial environment inside a deep space habitat. It would be a sealed system. Every microorganism inside the habitat would be something humans deliberately or accidentally brought aboard. The microbial diversity of Earth, estimated at potentially billions or more species across every ecosystem on the planet, would be reduced to whatever survived the journey and whatever the life support systems permitted. Air filtration, water recycling, waste processing, food production, every subsystem in a closed habitat manages and constrains the microbial ecosystem. The goal is to keep dangerous pathogens out and maintain a safe, stable environment. The unintended consequence is a microbial world impoverished beyond anything any human immune system has ever developed within. We already see the effects of this microbial impoverishment in astronaut health data. Studies of astronauts aboard the International Space Station have documented significant changes in gut microbiome composition during spaceflight. Beneficial bacterial populations decline. Anti-inflammatory species like lactobacillus and bifidobacterium decrease. Populations of stress-associated bacteria increase. Gut microbial diversity drops. The skin microbiome changes as well, with shifts that correlate with the high frequency of skin rashes and hypersensitivity reactions reported by astronauts. The immune system shows corresponding dysfunction. T-cell and B-cell counts drop. Natural killer cell function degrades. Latent viruses reactivate. Epstein-Barr virus, varicella zoster virus, and cytomegalovirus have all been detected reactivating in astronauts during or after spaceflight. Up to 50% of astronauts exhibit signs of immune deficiency upon returning to Earth. During the Apollo missions, more than half the astronauts became sick within a week of landing. These effects occur in adults with fully developed immune systems that trained on decades of Earth microbial exposure. Those immune systems have a vast library of immune memories to draw on. They have encountered thousands of pathogens and developed specific responses to each. Even in their degraded state, astronaut immune systems can call upon reserves of immunological experience that a spaceborne child would never possess. A child born and raised inside a sealed deep space habitat would develop an immune system trained on whatever microorganisms existed within that habitat. If the habitat contained a few hundred microbial species, rather than the millions a child would encounter on Earth, the immune system would calibrate itself to that limited world. It would learn to recognize and respond to those specific organisms. It would build immune memories for those specific antigens, and it would have no experience whatsoever with the overwhelming majority of microorganisms that exist on Earth. What would happen if that person traveled to Earth? They would be stepping into the most biologically diverse environment in the known universe with an immune system that has no training data for it. Common soil bacteria, that an Earth-raised child encounters before their first birthday would be entirely novel antigens to a space-raised immune system. Airborne fungal spores, pollen, animal dander, the microbial payload carried by every handshake and every breath of outdoor air, all of it would be foreign. The immune system might overreact, producing severe allergic or autoimmune responses to organisms that pose no threat to Earth-adapted humans. Or it might underreact, failing to mount effective defenses against pathogens that every Earth-raised child's immune system learned to handle in infancy. This is not a question of being sick for a few weeks while the body adjusts. Immune calibration during childhood development shapes the architecture of the immune system for life. The balance between inflammatory and anti-inflammatory responses, the ratio of different T-cell subtypes, the sensitivity thresholds for immune activation, these are shaped during childhood in ways that adult intervention can modify only at the margins. A person whose immune system developed in a sealed habitat might carry permanent immunological characteristics that make Earth's biological environment hostile to them in ways that no amount of post-arrival vaccination or medical treatment could fully correct. And the microbial problem extends in the other direction as well. Bacteria behave differently in space. Research has documented that certain bacterial species cultured in microgravity develop increased virulence, enhanced biofilm formation, and in some cases, specific antibiotic resistance. The closed environment of a space habitat, where the same microorganisms circulate through recycled air and water, creates conditions that could accelerate microbial evolution in unpredictable directions. Pathogens that are manageable on Earth might become more dangerous aboard a habitat, not because of any deliberate modification, but because the selective pressures inside a sealed, microgravity-influenced environment differ from those on Earth. For a child developing inside such an environment, the microbial world they are born into would shape their biology in ways we cannot fully predict. Their gut microbiome, their skin flora, their respiratory tract ecology, all would reflect the unique and impoverished microbial landscape of the habitat. Their immune system would be a product of that landscape, and their body's ability to tolerate Earth's microbial diversity would be untested and possibly inadequate. There is another biological system shaped entirely by environmental conditions during development, one that operates silently beneath every other process discussed so far. The endocrine system, the hormonal network that regulates growth, metabolism, stress response, sleep, puberty, and reproductive function, calibrates itself during childhood to the rhythms of the environment in which the child develops. And the most fundamental of those rhythms is the cycle of light and darkness produced by the rotation of the Earth. Every 24 hours, Earth turns once on its axis, creating a predictable alternation between daylight and night. This cycle is so ancient and so consistent that virtually every organism on the planet has internalized it. In humans, a cluster of neurons in the brain called the suprachiasmatic nucleus acts as the master biological clock, synchronizing the body's hormonal output to the light-dark cycle. When light enters the eyes, it signals the brain to suppress melatonin production and elevate cortisol, promoting wakefulness and alertness. When darkness falls, melatonin rises and cortisol drops, promoting sleep and cellular repair. Growth hormone, critical for childhood development, is secreted primarily during deep sleep. Thyroid hormones, which regulate metabolism and brain development, follow circadian patterns. Reproductive hormones that govern puberty are timed by the same clock. This circadian system is not hardwired at birth. It develops and calibrates during infancy and childhood through exposure to consistent environmental light cues. Newborns lack a mature circadian rhythm. Over the first months of life, regular exposure to daylight and darkness trains the suprachiasmatic nucleus to establish the 24-hour cycle that will govern hormonal regulation for the rest of that person's life. Disruption of this process has measurable consequences even on Earth. Research on shift workers has consistently demonstrated that chronic circadian misalignment is associated with menstrual irregularities, reduced fertility, increased rates of miscarriage, poor pregnancy outcomes, and metabolic dysfunction. Children of shift-working parents show altered sleep architecture and stress hormone profiles compared to children raised in households with conventional light-dark schedules. In a deep space habitat, there would be no natural light-dark cycle, no sunrise, no sunset, no seasonal variation in day length. The lighting environment would be entirely artificial, controlled by the habitat's electrical systems, and set to whatever schedule the engineers programmed. That schedule might approximate a 24-hour cycle, or it might not. The light itself would differ from sunlight in spectral composition, intensity, and the subtle variations that accompany weather, seasons, and the arc of the sun across the sky. A developing child would calibrate their circadian system to this artificial light environment. If the calibration is close enough to Earth's natural cycle, the endocrine system might develop normally. But if the artificial lighting lacks the full spectrum of natural sunlight, or if the schedule drifts from a true 24-hour period, or if the habitat's operational demands require variable lighting schedules, the child's hormonal calibration could drift with it. Growth hormone secretion patterns could shift. Melatonin production could become irregular. Cortisol rhythms could flatten, producing a chronic low-level stress response that the child experiences as normal because they've never known anything else. Puberty timing, which is hormonally regulated and environmentally sensitive, could accelerate or delay depending on the endocrine signals the body receives during the prepubertal years. These are not exotic risks. They are straightforward extrapolations from well-documented terrestrial research on circadian disruption. The difference is that on Earth, circadian misalignment is usually temporary and correctable. In a deep space habitat, it would be the permanent condition of the environment. A child would not be experiencing jet lag. They would be developing inside a world where the light-dark cycle that shaped human endocrine evolution does not naturally exist. Their entire hormonal architecture would reflect that world, and like every other system discussed in this story, that architecture, once established during development, does not easily recalibrate in adulthood. Beyond radiation and immune development lies a third category of vulnerability that compounds the first two: the total dependence of a spaceborne child on artificial life support systems. On Earth, the systems that sustain human life, atmosphere, water cycle, food web, temperature regulation, are planetary in scale and functionally limitless in redundancy. If one crop fails, others survive. If one water source is contaminated, alternatives exist. The atmosphere circulates and regenerates through photosynthesis across billions of plants and trillions of microorganisms. No single point of failure can collapse the system that sustains you. In a deep space habitat, every one of these functions must be performed by machines. Air must be scrubbed of carbon dioxide and replenished with oxygen by mechanical or biological recycling systems. Water must be recovered from urine, humidity, and waste through filtration and purification systems that cannot fail without immediate consequences. Food must be grown in hydroponic or aeroponic systems, or carried as preserved supplies with finite shelf life. Temperature must be regulated by thermal management systems that balance heat generation from equipment and human bodies against the frigid vacuum of space. Every one of these systems has failure modes. Every one requires maintenance, replacement parts, and energy to operate. A developing child is wholly dependent on these systems, not in the abstract, general way that all humans depend on their environment, but in the direct, immediate, machine-mediated way that a patient in an intensive care unit depends on their ventilator. If the atmospheric system fails, there is no stepping outside for fresh air. If the water recycling system degrades, there is no river to drink from. If the food production system suffers a blight or contamination, there is no grocery store, no neighbouring farm, no wild game to hunt. The margins are zero. Nutritional adequacy adds another layer of concern. Growing children require specific nutrients in specific quantities during specific developmental windows. Calcium and vitamin D for bone mineralization, iron for blood cell production, omega-3 fatty acids for neural development, iodine for thyroid function, zinc for immune maturation. On Earth, these nutrients are available through a diverse food supply drawn from thousands of plant and animal species. In a space habitat, the food supply would be limited to whatever could be grown or stored aboard. Nutritional deficiencies during critical growth periods can cause permanent developmental damage that no later supplementation can fully reverse. A habitat food system would need to provide complete, balanced nutrition for every stage of childhood development, from neonatal feeding through adolescence, without interruption, without shortage, without error, for the entire duration of the child's growth. Medical care in deep space introduces another dimension of risk. On Earth, pregnancy and childhood development are supported by an enormous medical infrastructure. Prenatal monitoring, ultrasound imaging, blood testing, emergency obstetric surgery, neonatal intensive care, pediatric specialists, vaccination programs, dental care, orthopedic intervention, all of these exist because human development is complex enough to require them. In a deep space habitat, medical capabilities would be limited to whatever equipment, medications, and expertise traveled with the crew. If a child developed a congenital heart defect that required surgical correction, the surgery would need to happen aboard the habitat or not at all. If a premature delivery required neonatal intensive care, that care would need to be available with the equipment on hand. If a childhood illness required a specialist that the crew did not include, the child would go without. The cumulative picture that emerges from these compounding vulnerabilities is sobering. A child born in deep space would develop under altered gravity, elevated radiation exposure, a reduced and potentially atypical microbial environment, nutritional constraints imposed by a limited food supply, and total dependence on artificial systems with no terrestrial backup. Each of these factors individually introduces developmental uncertainty. Together, they create a developmental environment so different from Earth's that the body produced by it may differ from an Earth-developed body in ways we cannot fully anticipate. And this brings us to the deepest tension in the entire conversation about deep space human settlement. Every one of these problems has a potential technological mitigation. Rotating habitats could provide artificial gravity. Hydrogen-rich shielding and magnetic fields could reduce radiation doses. Carefully managed microbial ecosystems could diversify the habitat's biological environment. Advanced agricultural systems could ensure nutritional completeness. Telemedicine and artificial intelligence could extend medical capabilities. None of these solutions are impossible. Some are being actively developed, but none of them are guaranteed to be sufficient. The question is not whether humanity can build better technology. The question is whether better technology can replicate the developmental environment of Earth closely enough that a human body develops as if it were on Earth. The margin for error is not defined by engineering tolerances. It is defined by the sensitivity of biological development to environmental conditions, a sensitivity honed by hundreds of millions of years of evolution under one specific set of physical parameters. A rotating habitat might produce 95% of Earth's gravitational signal. But if the remaining 5% includes the Coriolis distortion and gravity gradient effects that alter vestibular calibration during the critical fetal period, that 5% gap could translate into permanent developmental differences. The same logic applies to radiation shielding that blocks 90% of galactic cosmic rays but allows 10% through during the first trimester, to an immune training environment that contains 1,000 microbial species instead of 1,000,000, to a nutritional supply that meets 99% of developmental needs but lacks a trace mineral essential for a process we have not yet identified. Biology does not grade on a curve. It does not award partial credit for almost replicating the conditions of Earth. It responds to the conditions that actually exist, and it builds accordingly. And there is one more dimension to this problem that most discussions of space settlement never address. Everything described so far considers a single generation. The first children born in deep space. But those children, if they survive and thrive in their habitat, will eventually have children of their own. And the bodies of those second-generation space-born humans will develop not inside Earth-formed mothers, but inside mothers whose own bodies were shaped by the habitat environment. This is where the developmental divergence from Earth stops being a single event and becomes a compounding process. Consider what this means in concrete biological terms. A woman whose skeleton developed in fractional gravity carries lower bone mineral density than a woman whose skeleton developed on Earth. During pregnancy, the mother's body supplies calcium and other minerals to the developing fetal skeleton. If the mother's bones contain less mineral to begin with, the biochemical environment available to the fetus may differ. The mechanical forces acting on the fetus in utero depend partly on the mother's posture, movement patterns, and gravitational environment, all of which would reflect the habitat rather than Earth. The hormonal signals that regulate fetal growth pass through a maternal endocrine system that itself calibrated to an artificial light cycle and a low-gravity environment. The microbial species that colonize the infant during and after birth come from the mother's body, which hosts a microbiome shaped by the sealed habitat ecosystem. Each of these factors means that the second generation does not start from the same biological baseline as the first. The first generation developed from Earth-formed parents. Their bodies diverged from the Earth baseline because they grew in a different environment, but they began the journey from an Earth starting point. The second generation begins from a starting point that is already shifted. Their parents' bodies are the developmental environment, and those bodies have already adapted to the habitat. The divergence does not reset, it compounds. This compounding is not genetic. No mutations need to occur. No evolutionary selection is required. It operates entirely through developmental biology, the same process that has been the subject of this entire discussion. Each generation develops in response to the conditions it actually experiences, and those conditions include the bodies of the parents, which were themselves developmental products of the previous generation's environment. A child developing inside a mother whose cardiovascular system is calibrated to low gravity is developing inside a different hemodynamic environment than a child developing inside an Earth-calibrated mother. The signals are different, the nutrients may be partitioned differently, the mechanical forces are different, and the child's body responds accordingly. Over 3 or 4 generations, the cumulative effect could be substantial, not because any single generation experiences a dramatic deviation from the one before it, but because each generation's starting point is the previous generation's endpoint. A photograph copied from a copy, each iteration faithfully reproducing what it receives, but each reproduction slightly further from the original. No single copy introduces a glaring error, but after enough generations, the image is recognizably different from the first print. This generational compounding means that the question of whether spaceborne humans can survive Earth is not static. It is not a question that gets answered once by the first generation and then holds forever. The first generation, born to Earth-developed parents, might retain enough developmental similarity to Earth-standard human biology that a difficult return to Earth remains physically possible. Their bones might be thinner but not dangerously so. Their hearts might be smaller but not critically so. Their immune systems might be naive but not helplessly so. The fifth generation might be a different story entirely. 5 successive rounds of development in habitat conditions, each building on the biological baseline of the generation before, could produce a body so thoroughly calibrated to the artificial environment that Earth compatibility is no longer a realistic question. Not because anything went wrong, because everything went exactly as biology dictates. Each generation adapted faithfully to the world it knew, and after enough faithful adaptations, the distance between that world and Earth became too great for a single human body to bridge. This multi-generational trajectory is perhaps the most sobering aspect of the entire subject. It means that the decision to establish a permanent human population in deep space is not merely a decision about the people who go. It is a decision about every generation that follows. The first settlers choose to leave Earth. Their grandchildren may discover that the choice was made for them in a far more permanent sense than anyone anticipated. Not because they were denied passage home, because their bodies, built by the only world they have ever known, made the passage itself survivable only in theory. Every one of these factors shapes the body of a developing child. Gravity shapes the skeleton, the muscles, the heart, the vestibular system. Radiation shapes the integrity of the genome. Microbial exposure shapes the immune system. Nutrition shapes every tissue. Medical availability determines whether developmental complications are correctable or permanent. Together, these forces do not simply influence what a deep space child looks like. They determine what that child's body can do, what forces it can withstand, what environments it can tolerate, what world it belongs to, and that is where this story arrives at its most unsettling destination. Because everything described so far, every risk, every uncertainty, every developmental variable, leads to a single, unavoidable question. If a human being develops under all of these altered conditions, if their skeleton, muscles, heart, inner ear, immune system, and organ function all calibrate to an artificial habitat in deep space, what happens when they encounter the world that built every other human who has ever lived? What happens when a body shaped by a rotating drum and recycled air and limited microbes and reduced radiation shielding meets a planet with crushing gravity, a raging biosphere, unshielded sunlight, and an atmosphere denser than anything they have ever breathed? What happens when a child of deep space tries to come home to a planet that was never theirs? Picture the moment. A ship returns to Earth orbit after a journey that lasted years or decades. Inside that ship is a person who has never set foot on this planet. They were born aboard a rotating habitat somewhere in the outer solar system or beyond. They grew up walking on curved floors, breathing recycled air, drinking water that had been filtered through the same purification system 1,000 times. They learned to run and play in a world where the gravitational pull was a fraction of what waits below. Their bones formed under that fraction, their heart calibrated to it, their inner ear tuned itself to the particular version of down that a spinning drum produces. Now the ship is in orbit. Below them, blue and white and vast beyond anything they have ever seen, sits the planet their species came from. Everyone aboard has heard the stories. Earth, the origin, the cradle, every song, every myth, every piece of history that travelled with them across the void points back to that sphere of rock and water and air. It is the most important place in the collective memory of the human species, and for the person who was born in space, it may also be the most dangerous place they could ever go. This is not a dramatic exaggeration. It is the logical conclusion of everything the science tells us. If a human body develops under different gravitational, radiological, microbial, and atmospheric conditions, that body will be optimized for those conditions, not for Earth's. And the gap between the conditions of a deep space habitat and the conditions on the surface of this planet is not a gentle slope, it is a cliff. Start with the most immediate and visceral challenge: gravity. The moment a space-developed person steps onto Earth's surface, their body would experience a gravitational force potentially several times stronger than anything they grew up with. If their habitat provided 0.3 g through rotation, Earth would feel more than 3 times heavier. If it provided 0.16 g, roughly lunar gravity, Earth would feel 6 times heavier. Every kilogram of their body weight would multiply. Their own skeleton, muscles, and organs would suddenly weigh far more than the structures supporting them were ever built to handle. For an adult astronaut returning from 6 months in microgravity, standing upright on Earth is difficult but achievable within days or weeks. Their bones lost density, their muscles atrophied, their cardiovascular system deconditioned, but these systems were built for 1G and can readapt because the architecture is already there. The blueprint was executed decades ago. The recovery is a return to a known state. For a person whose skeleton never developed under 1G, there is no known state to return to. Their bones are not weakened versions of Earth-standard bones. They are correctly built bones for a lower gravity world. The mineral density, the trabecular architecture, the cortical thickness, all reflect the mechanical loads they actually experience during growth. Placing those bones under Earth's full gravitational load would be like loading a bridge with 3 times the weight it was engineered to carry. The structure might hold for a while, it might not. Stress fractures in the spine, pelvis, and legs could begin within hours or days. Weight-bearing joints designed for lighter loads could degrade rapidly. The simple act of standing could become a medical event. The muscles tell a similar story. Postural muscles that never developed. The fiber density and neural recruitment patterns for 1G would fatigue rapidly under Earth's pull. Walking would be exhausting. Running might be impossible. The core muscles that stabilize the spine during upright movement, muscles that Earth-raised children develop through years of crawling, climbing, and playing against gravitational resistance, might lack the endurance to keep the spine aligned under its own weight. The body would not simply feel heavy, it would feel like it was collapsing under itself. The cardiovascular consequences could be even more immediate and more dangerous. A heart that developed in low gravity would be smaller, with thinner ventricular walls and lower stroke volume than a heart that grew under 1G. On Earth, that heart would need to pump blood upward against a hydrostatic gradient far steeper than anything it was designed for. Blood would pool in the lower extremities. Blood pressure to the brain would drop. The baroreceptors, those pressure sensors calibrated to a low-gravity circulatory environment, would be overwhelmed by pressure differentials they were never trained to manage. Orthostatic intolerance plagues astronauts for days or weeks after missions, lasting mere months, even though their hearts spent decades building strength under 1G before they ever launched. For a person whose entire cardiovascular development occurred in low gravity, this would not be a temporary symptom. It might be a permanent condition. Standing upright on Earth for more than a few minutes might cause them to lose consciousness. Every time, the vestibular system would face its own crisis. Everything we know about vestibular development indicates that the inner ear calibrates its sensitivity and its neural integration to the gravitational field present during fetal life and childhood. A person whose vestibular system developed under a rotational simulation of partial gravity would have a sense of balance tuned to that specific environment. The Coriolis effects, the gravity gradients, the particular way that simulated gravity behaves during head movements inside a rotating habitat, all of these would be encoded into their vestibular processing as normal. Earth's gravity would feel fundamentally wrong. Not just stronger, different in character, uniform rather than gradient dependent, free of Coriolis distortion, pulling in one consistent direction rather than varying with position relative to the axis of rotation. The result could be chronic, unrelenting vertigo, a sensation of falling that never resolves because the brain's model of what gravity should feel like does not match what it is experiencing. Nausea, spatial disorientation so severe that navigating a room becomes impossible without assistance. Inability to stabilize gaze during head movement because the vestibulo-ocular reflex, the automatic eye movement that keeps your vision steady when your head turns, calibrated to a different gravitational input. This is not an adjustment period that resolves in weeks. Vestibular calibration occurs during developmental windows that close in childhood and adolescence. An adult whose vestibular system calibrated to a non-Earth environment may never fully adapt to Earth's gravitational field because the neural architecture that would enable that adaptation was finalized years ago. Then come the eyes. If SANS can reshape the eyeball and compromise the optic nerve in adults who spent decades developing under Earth's gravity, what would happen to eyes and optic nerves that formed from the embryonic stage under entirely different fluid dynamics? The intracranial pressure environment during fetal development influences how the skull forms, how the brain is cushioned, how cerebrospinal fluid circulates, and how the optic nerves are bathed and supported. A space-developed cranial anatomy might differ in ways that function perfectly in the habitat, but create immediate problems under Earth's gravity. Fluid that distributed evenly in microgravity would pool differently under 1G, pressing on structures that were never shaped to bear that pressure. Vision changes that are gradual and partially reversible in astronauts could be acute and permanent in someone whose anatomy assumed those fluid dynamics from the start. And then there is the invisible threat, the one that carries no weight and makes no sound, the biosphere. Earth is the most biologically dense environment in the known universe. Every surface is colonized. Every breath of outdoor air contains thousands of microbial species. Soil teems with bacteria, fungi, protists, and viruses in concentrations that would be unimaginable inside a sealed habitat. The oceans are microbial ecosystems of staggering complexity. The oceans, even the human beings walking the streets, carry trillions of microorganisms on their skin, in their mouths, in their intestines, each person a walking ecosystem unto themselves. A spaceborne human arriving on Earth would be stepping into this biological torrent with an immune system that has never encountered any of it. Their immune training occurred inside a sealed container with a managed microbial population numbering in the hundreds or low thousands of species. Earth's microbial diversity exceeds that by orders of magnitude. The immune system responds to novelty with inflammation. Novel antigens, substances the immune system has never seen before, trigger defensive responses. A small number of novel encounters is manageable. The body investigates, categorizes, and either mounts a targeted response or develops tolerance. But a massive, simultaneous exposure to thousands of unfamiliar microbial species could overwhelm an immune system with no training data for any of them. The consequences could range from severe allergic reactions to runaway inflammatory responses to vulnerability to infections that Earth-raised humans shrug off in childhood. Common respiratory viruses, skin bacteria, foodborne organisms, environmental fungi, all would be first contact pathogens for a space-developed immune system. Even the beneficial microorganisms that colonize the gut and skin of Earth-raised humans would be foreign to a body whose microbiome developed in a radically different microbial context. The simple act of shaking hands with an Earth-born human could introduce microbial species that a space-born person's body has no framework for tolerating. This is not the same as an astronaut getting a cold after landing. Astronauts who fall ill after returning from space are experiencing a temporary immune suppression that resolves as their bodies readapt. Their immune systems were trained on Earth. They have decades of immunological memory to draw on. They know these pathogens. They are temporarily weakened, not immunologically naive. A space-born person would be genuinely naive. Their immune system would be encountering Earth's microbial world for the first time, with no prior exposure, no childhood training, no accumulated immune memory. The analogy is not a traveler catching a foreign bug. The analogy is a deep-sea fish brought to the surface. The environment it was perfectly designed for is the only environment it can survive. Now compound all of these factors simultaneously. A skeleton too fragile for 1G, muscles too weak to support upright posture, a heart too small to maintain blood pressure against full gravitational pull, a vestibular system calibrated to the wrong version of down, eyes and cranial structures formed under different fluid dynamics, an immune system with no training data for Earth's biosphere, lungs accustomed to a controlled atmospheric composition, suddenly breathing an atmosphere at full sea level density with an uncontrolled mix of particulates, allergens, and microorganisms. A body that functions perfectly in the world that built it, encountering a planet that built every other human but not this one. This is not instant death. It is important to be precise about that. A space-born human who travelled to Earth would not step off a spacecraft and collapse. The effects would be graduated, overlapping, compounding. Some would manifest in hours. Orthostatic intolerance, vestibular disorientation, muscular fatigue. Others would develop over days or weeks. Stress fractures, immune reactions, cardiovascular strain. Still others might emerge over months or years. Joint degradation under chronic gravitational overload, progressive skeletal failure, chronic inflammatory conditions driven by continuous novel immune encounters. The trajectory would depend on how different their developmental environment was from Earth's, and how much their body could compensate through plasticity and adaptation. But the critical point is that adaptation has limits, and those limits are set during development. An adult brain retains some neuroplasticity, some ability to rewire in response to new inputs. But the foundational architecture, the calibration of the vestibular system, the baseline cardiovascular capacity, the structural density of the skeleton, these are established during growth and do not fully reset in adulthood. A spaceborne person might adapt partially to Earth. They might learn to tolerate some aspects of the environment with medical support, physical therapy, and pharmaceutical intervention. But it is scientifically plausible that full adaptation might never occur. That Earth would remain, for the rest of their life, a planet that their body was not built for. And here is where the ethical dimension becomes unavoidable. No child has ever been born on Earth under conditions known to carry this degree of developmental uncertainty. Every human society has norms, laws, and ethical frameworks that protect children from known risks during their development. We do not expose pregnant women to radiation if we can avoid it. We do not deliberately deprive developing children of essential nutrients. We do not conduct medical experiments on infants without compelling justification and rigorous oversight. But a child conceived and born in deep space would be, by definition, a developmental experiment. An experiment in which the subject cannot consent. An experiment in which the variables, gravity, radiation, microbial environment, atmospheric composition, nutritional constraints, medical limitations, are imposed by the circumstances of the parent's choice to live in deep space. The child does not choose to be born aboard a rotating habitat. The child does not choose to develop under fractional gravity. The child does not choose to grow an immune system trained on a sealed ecosystem with 1,000th of Earth's microbial diversity. These conditions are inherited, not chosen. And their consequences, if they include permanent physical divergence from Earth-viable human development, are irreversible. This does not mean that deep space reproduction is inherently unethical. Humans have always raised children in environments that carried risk. Frontier settlements, polar outposts, high-altitude communities all imposed developmental conditions that differed from the species norm. Children born at high altitude developed larger lung capacity and higher red blood cell counts. Children raised in equatorial climates developed different thermoregulatory responses than children raised in Arctic environments. Human development is plastic. It adapts. The question is whether the degree of environmental difference in deep space exceeds the range of plasticity that human development can accommodate without producing bodies that can no longer function in the ancestral environment. We do not know the answer. That uncertainty is itself the ethical problem. We are contemplating raising children under conditions whose developmental consequences are largely unknown, in an environment from which return to the ancestral planet may be physically impossible for the children thus raised. The parents chose deep space. The children inherit its consequences, and if those consequences include permanent physical adaptation to the habitat environment, then something profound has happened. Something that goes beyond medicine or engineering or even ethics. Something that touches the definition of what Earth means for the species that came from it. For every generation of human beings that has ever lived, Earth was home. Not in the sentimental sense, though it was that too. In the physical sense, the literal, biological, gravitational, atmospheric, microbial sense. Every human body was a product of this planet. Every skeleton was shaped by its gravity. Every immune system was trained by its biosphere. Every heart was strengthened by its pull. Earth was not just where humans lived. It was what humans were made of and made by. A child born in deep space would be the first human in the history of the species for whom that is no longer true. Their body would be a product of a different world. A world built by human engineers rather than by geological and biological processes spanning billions of years. A world with its own gravity, its own atmosphere, its own microbial ecology, its own constraints and parameters. That child would belong to that world in the same way you belong to Earth. Not by choice but by development. Not by identity but by biology. Their bones would be its bones. Their heart would be its heart. Their sense of balance, their immune tolerance, their muscular capacity, all would be calibrated to the habitat that raised them. And Earth, the planet that gave rise to Homo sapiens, the world that shaped every ancestor in an unbroken chain stretching back hundreds of thousands of years, would become something else for that child. Not home. Not origin. Not even a place they could safely visit. Earth would become an ancestral world, a place of stories and history and cultural memory that their body cannot inhabit. A planet their species came from, but that they, personally, were not built to survive. This is the most haunting implication of everything the science tells us. Not that spaceborne humans would be less human. They would not be. They would carry the same genome, the same cognitive capacity, the same emotional depth, the same capacity for love and curiosity and wonder as any human born on Earth. They would be fully, completely, unambiguously human, but they would be human in a way that no human has ever been before. Human in a body shaped by an artificial world, rather than a natural one. Human in a form that belongs to a place humanity built rather than the place that built humanity. The disturbing truth is not mutation. It is not deformity. It is not the creation of a subspecies or an alien. It is the possibility that the ordinary, natural, perfectly functional process of human development, running exactly as evolution designed it to run, could produce a body so adapted to an artificial environment that the planet of origin becomes hostile territory. Not through any failure of the body, through the body doing precisely what it has always done, adapting to the world it grows up in, building itself for the conditions it experiences, becoming what its environment tells it to become. Earth would still be there, blue and bright and beautiful, visible from orbit, reachable in days or weeks depending on where in the solar system the habitat drifts. Every piece of human culture, every language, every work of art, every scientific discovery, every spiritual tradition would point back toward it. The spaceborne child would know Earth the way a descendant knows the country their great-grandparents emigrated from, through photographs and stories and a longing that has no physical correlate. They could look down at it from orbit. They could study its geography and weather and ecosystems through telescopes and data feeds, but stepping onto its surface might be a language their mouth was never shaped to speak, a place their body understands only as a concept, not as a livable environment. And this would not be a tragedy of technology failing. It would be a consequence of technology succeeding. The habitat works, the life support works, the artificial gravity works well enough to sustain a population. Children are born, grow, thrive, live full lives, have children of their own. The deep space settlement succeeds at everything it was designed to do, and in succeeding, it creates human beings for whom the world of their ancestors is no longer a viable destination. The species that left Earth to ensure its survival would produce descendants who can never return. This is what it means to say that the first humans born in deep space may never survive Earth. Not that they would die on contact. Not that they would be broken or damaged or inferior, but that their bodies, shaped by the world humanity built for them rather than the world that built humanity, might find Earth's gravity too crushing, its biosphere too foreign, its atmosphere too dense, its demands too extreme for a form that was never constructed to meet them. Earth would remain the origin of the species, but it would no longer be a survivable home for every member of it. We stand at the edge of that possibility right now, not in the distant future, not in the realm of pure speculation. In the scientific present, where every piece of evidence about human development, spaceflight physiology, radiation biology, and immune function points toward a single conclusion, that the human body is a product of its environment in the deepest, most literal sense, that growing up somewhere other than Earth means becoming something shaped by somewhere other than Earth, and that the planet which gave us everything, the gravity that built our bones, the air that filled our lungs, the microbial world that trained our defenses, the magnetic field that shielded our cells, may one day look down at children carrying human DNA and human minds and human hearts and see strangers, not because those children chose to be different, because they were built by a different world, a world we made. The greatest irony of human space exploration may turn out to be this: we left Earth to preserve the species, and in doing so, we may create members of the species for whom Earth is no longer a place of preservation. It is a place of peril, the cradle that rocked us into existence, waiting at the bottom of a gravity well that our deepest descendants may never be able to climb down into and survive. That is the question this generation must begin to answer. Not because the first deep space birth is happening tomorrow, but because every year we move closer to the moment when it becomes possible. And when that moment arrives, the child who is born will not get to choose what world their body belongs to. That choice will already have been made by the gravity they develop under, by the radiation they absorb, by the air they breathe and the microbes they encounter, and the forces that press on every cell as it divides and differentiates and builds the only body they will ever have. We owe that child, wherever and whenever they are born, the honesty to admit what we do not know, and the courage to find out before we ask a human body to become something Earth may no longer recognize as its own.